everyone, welcome to our channel Love of Biology. Today we are going to learn about carbohydrates. Before that, please subscribe my channel. And carbohydrates are the sugars and the polymers of the sugars. The simplet carbohydrates are monosaccharides, and macromolecules we are calling them as polysaccharides. So that is polymers composed of many sugars. Saccharide means sugar here, so sugars will form the building blocks, those two monosaccharides. So there are three types of the carbohydrates: monosaccharides, disaccharides and polysaccharides. So monosaccharides means mono means single, so they made up of single sugar, example glucose, ribose, etc. Disaccharide, di means a single two and saccharides means sugar, so they are made up of two sugars which are linked by glycosidic bonds, examples lactose, sucrose. And polysaccharides made up of many sugar molecules to form the long chain. So examples are many: starch, cellulose, chitins, peptidoglycans. So those are nothing but the cell walls, right? So monosaccharides have molecular formulas that are usually multiples of C, carbon, hydrogen and oxygen. Glucose is the most common type of the monosaccharide. Actually, these are also divided into few types based upon their location of carbonyl groups and number of carbons in the carbon skeleton. Let us see those types of the monosaccharides based upon the carbon number: trioses, tetroses, pentoses, hexoses and heptoses. Trioses means 3 carbons containing sugar molecule. Glyceraldehyde is example. Tetroses means obviously it contains 4 carbons, example erythrose. And pentose means sugars with 5 carbons, right? So example deoxyribose and ribose. Hexoses are the normal glucose with the 6 carbons, and heptoses with the 7, pseudoheptulase example. Okay, monosaccharides serve as a major fuel for the cell and as raw material for building molecules. Actually, they will provide the energy. They are the energy sources. Disaccharide means they are formed when a dehydration reaction joins two monosaccharides with the help of a bond glycosidic linkage. They will attach. Lactose and sucrose are the examples. Lactose, it contains 2 sugars because it is a disaccharide. They are galactose plus glucose, whereas the sucrose, table sugar, that is glucose and fructose. Fructose is a fruit sugar. They will combine to form the sucrose. Polysaccharides, polymers of the sugars, have storage and structural roles. The structure and function of the polysaccharides are determined by its sugar monomers and the position of the glycosidic linkages. So these polysaccharides actually based upon the type of the sugar molecule present in their chain. They are divided into homopolymers as well as heteropolymers. Okay, so these will form the storage as well as structural molecules. So let us see the what are heteropolymers and what are homopolymers. Homopolymer means which contains the same type of the sugar. The complete chain made up of the same type of the sugar. Whereas the heteropolymer means they are made up of different types of the sugar. So few examples: storage polysaccharides, starch and glycogen. Starch is a storage polysaccharide of the plants, right? It is a homopolysaccharide made up of same type, that is only glucose monomers it is made up of. Plants store starch as granules within the chloroplast and the other plastids, right? And glycogen is a storage form of sugar in the animals. It is also a homopolymer. The structural polysaccharides, if you see, cellulose is a major component of the tough wall of plant cells. Like starch, cellulose is a polymer of glucose, but the glycosidic linkages differ. The difference is based upon the 2 ring forms, alpha and the beta of glucose. Polymers with the alpha glucose are helical. They will form the helical structure. Polymers with beta glucose are straight chain containing 1. So in straight structures, hydrogen atoms on one strand can bond with OH groups on the other strand. Parallel cellulose molecules held together with where they are grouped into microfibrils which form the strong building molecules. So here you can see the structure of starch as well as the cellulose. So because of this structure, so starch will will absorb the iodine in the experiment, right? Because of the structure. Polymers with alpha glucose are helical and polymers with beta glucose are straight. Already we learned this, right? So that is the thing here. Because of this, the structure. Enzymes that digest the starch by hydrolyzing the alpha linkages can't hydrolyze the beta linkages in the cellulose. So cellulose in the human food passes through the digestive tract as insoluble fiber, and some microbes use enzymes to digest the cellulose. Many herbivores form from cows to the termites have symbiotic relationship with these microbes. Because of this structure, cellulose is not digested in our body because their structure. So this is important. And other one is chitin. So another structural polysaccharide, it is found in the exoskeleton of the arthropods as well as the fungus, right? So fungus cell wall is also made up of chitin. It also provides structural support for the cell walls of the fungi. Thank you for watching. Please subscribe.